Hi, I am Thien Thien. I'm the social media editor of the Parkinsonism and Related Disorders journal. And today we are fortunate to speak with Drs. Henry Mauricio and Nacho, authors of a thought-provoking editorial published in the January 2026 PRD journal entitled Is One Too Many? Rethinking the Innocence of Heterozygous Variants in Recessive Disorders. Thank you for being here today.
Thank you very much for the invitation.
We understand the classical Mendelian principles of autosomal dominant and autosomal recessive inheritance. So what are the limitations and challenges of applying these principles to neurodegenerative disorders such as Parkinson's disease?
Yeah, so you can imagine that this, classical Mendelian inheritance works well with diseases that are really caused by like one single really high penetrant gene. And Parkinson's disease, for the last 20 years, we've shown that is not the case, right? So there's, over 20 familial genes, over 150 risk genes. And also even the genes that we think cause disease are not fully penetrant. So most of the variants, don't cause the disease 100% of the time. That's what the penetrance means, right? So even if you carry the variant, it doesn't mean that you will develop the disease. We also see a lot of heterogeneity in terms of the age of onset, in terms of the clinical presentation, both motor and non-motor symptoms. So all this makes the genetic profile or the genetic component of the disease is very complex. And then you can also add that there's environmental factors, there's gene-environment interactions. So a model where we think about use one variant causing the disease or two variants in the case of recessive doesn't work well with with Parkinson's disease, and it makes it hard to interpret the results. And I think as we sequence and we genotype more samples, we're realizing really how complex this enterprise is. Yeah.
Very interesting and very confusing to some of us as well. So how do heterozygous pathogenic variants contribute to disease risk and phenotypic expressions in certain Parkinson's disease-associated genetic disorders?
Yeah, so one of the first things that, when we were writing this, we realized is that the first thing that we need to take into consideration is that a lot of the times we think that a person only carry one heterozygous variant in one recessive gene, which again, you would expect that that person would not develop the disease just because you usually for a recessive disease or gene, you need two variants. And the first thing that we need to realize is that a lot of the times there might be a second variant that we never found because variants come in different flavors and different shapes. Single nucleotide variants are very easy to identify. Copy number variants are a little bit harder to identify, but there's also inversions. There's also all kinds of very complex variant. So the first thing is to make sure that that person really carry only one heterozygous variant, and it's a really good example that we included in our article in the Parkin gene, which is a recessive, probably the most important recessive gene in Parkinson's disease. In a Japanese two siblings of early onset Parkinson's disease in Japan, where there was a copy number variant in one of the chromosomes, and they thought that these individuals were just heterozygous for Parkin. But then when they did long read sequencing, so it required a technique that usually we don't do in all patients, which is long read sequencing, where you sequence the whole gene in one piece, not multiple small pieces, and then try to match them together. They identified that there was an inversion within the gene on the other allele. So this person was not really a heterozygous; it was actually a compound heterozygous. So there's two variants. So that's the first thing that we need to address in terms of heterozygous variants. But the other thing is that there are truly heterozygous variants that sometimes can modify the phenotype. And the way they do this is usually heterozygous variants in recessive diseases cause loss of function. So you can imagine that there is a partial loss of function of that gene, not a complete loss of function, which might not be enough to cause the disease, but it could cause or it could modify the phenotype of that individual. And you can also think that because we have so many genes involved in Parkinson's disease, there could be multiple hits in the same pathway, right? So, for example, in the paper that we wrote the editorial about, what they saw is that some of these heterozygous carriers, there were also carriers for variants in other Parkinson's genes, and a lot of them targeted the same mitochondrial lysosomal pathway. So that means that, again, is the accumulation of several variants at the end of the day what makes some people develop the disease. And also because you have different genes, then you have different ways that the disease might show up. And you will see, for example, in this case, some of the clinical symptoms of one of the genes showing up in another a genetic form of Parkinson's disease, for example, LERK2 and BPS 13C, like we see in this article. So again, it's just showing how complex these interactions are, and we have to think about this in the whole system, not in like one single gene, one single protein, but how all those really interact with each other and what the damage at the end of the day can be when you, for example, have multiple hits.
Very interesting. Well, Malfer et al. Published a paper, a research article in the January 2026 PRD entitled VPS 13C, heterozygous loss of function as modifiers of suboptimal levodopa response in Parkinson's disease. Could you elaborate on the key findings and their clinical implications?
Yeah, so I will take this one, Thien Thien. So absolutely, in this month's edition of Parkinsonism and Related Disorders, Malfer and colleagues, they publish a series of 4 cases of people with Parkinson's disease who shared, I will say, 2 main characteristics. The first involved clinical characteristics. Specifically, all of them, they had a early disease onset. They had a poor response to levodopa treatment despite an initially good response, and they also presented non-motor symptoms along with cognitive declinement. The second shared characteristic, and perhaps the most interesting finding, was genetic. As Nacho already mentioned, these individuals were carriers of heterozygous variants, pathogenic variants in VPS 13C, along with other variants in other Parkinson's disease genes. This is particularly interesting because we currently know that Parkinson's disease caused by VPS 13C follows a autosomal recessive inheritance pattern, meaning that both copies of the gene need to be damaged or mutated. However, as I just mentioned, the patients reported by Malfer and colleagues, they only had one allele mutated. And so in theory, this is not enough to develop the disease. So the hypothesis that they proposed, also already briefly mentioned by Nacho, is that these heterozygous pathogenic variants could act as modifiers of the phenotype, meaning that they can modify the presentation of Parkinson's disease, but not cause the disease by their own. And how this interacts, so Nacho also mentioned these participants, they also had variants in this gene, but also variants in other genes. And that's how the hypothesis that they propose comes up, regarding that it probably is following a multiple hit or shared pathway hypothesis.
I must say I learned something new from this paper. I know you mentioned a few, but what are the typical clinical features associated with these pathogenic variants in VPS 13C? Perhaps you could summarize it for us?
Yeah, absolutely. So important to mention is that the main clinical characteristics or features of homozygous carriers, and I want to highlight these homozygous carriers, are early disease onset, meaning that the disease presents usually before the age of 50, a rapid and severe disease progression, early cognitive declinement. They can also present dystonic features, and motor fluctuations and levodopa-induced dyskinesias are common. The important thing here, again, I just want to highlight this again, is that if you compare these characteristics with those reported by Malfer et al., you'll find that they are very similar to the features observed in their cases, even though they were heterozygous carriers of the variants. So that's why it's very interesting. Despite they don't have both alleles mutated, they are presenting these clinical characteristics.
And how is the VPS13C thought to be implicated in the pathogenesis of Parkinson's disease?
Yeah, this is a great question, and Nacho already mentioned a little bit about the importance of BPS13C and the function of mitochondria. But to wrap this again, so mitochondria and lysosomes are both two very important cellular organelles that are closely related with the pathogenesis of Parkinson's disease. So what happens here is that VPS13C, it's in charge of the homeostasis of the mitochondria, which means taking care of a correct function of the organelle. And to do this, it works very close with other proteins such as PINK1 and Parkin that we know that are also very important, relevant proteins and genes in Parkinson's disease pathogenesis. So when VPS13C stops functioning, the membrane potential of the mitochondria it's lower, and this upregulates a process known as PINK1 Parkin dependent mitophagy, which means that the mitochondria start to be targeted and degraded and cleared from the cell. And if you start to remove all the mitochondria from the cell, this is gonna lead to the presentation of a disease. But also, I also mentioned lysosomes that are important in this process. So what we've seen is that by VPS13C, also it's important for lysosomal function. And when VPS13C is damaged, the function, the distribution, and the volume of lysosomes are also affected. And affecting this volume can lead, for example, to the mitochondrial DNA material to leakage to the cytosol of the cell. And this DNA just moving around creates some neuroinflammatory response. And this neuroinflammatory response is finally what it's linked to Parkinson's disease pathogenesis. So this is what we know so far, how VPS13C variants are related with Parkinson's disease.
Thank you once again for sharing these wonderful insights, and the editorial challenges us to rethink the long-held genetic assumptions and highlights heterozygous variants still carry important clinical consequences. So to all our readers, and please do read the PRD editorial entitled "Is One Too Many? Rethinking the Innocence of Heterozygous Variants in Recessive Disorders," and also the research article by Malfer et al. Entitled VPS13C Heterozygous Loss of Functions as Modifiers of Suboptimal Levodopa Response in Parkinson's Disease. Thank you very much.
Thank you, Thien Thien.
Thank you.